Welcome to Biotanemy.
Ribosomal display.
Ribosomal display is an in vitro technique used to identify and study proteins with specific binding properties. That is, ribosomal display come in handy if you want to screen a specific protein synthesized by a specific gene, but you don't want to insert them into any cells or virus for the genome to express the protein. This system allows us to directly investigate peptide-protein interactions, DNA-protein interactions, or to select the peptide with desired characteristics without the application of living cells. The ribosomal system works by translating mRNA into peptides while maintaining the physical linkage between the ribosomes, mRNA, and synthesized peptide. The ribosomal display begins by translating the mRNA, as the ribosomes translate mRNA to synthesize the peptide, and we keep the ribosome to remain bound to the mRNA. This can be done by 1. Adding ribosome stalling sequence. Certain sequences or secondary structures in the mRNA is designed to cause the ribosome to stall or remain bound upon completing the translation. 2. By the addition of C-terminal tags. For example, SECM stalling sequence. The specific amino acid sequence within SECM that causes ribosomal stalling is often a hydrophobic segment followed by a series of residues that promote the pausing of translation. In many cases, this sequence includes a stretch of hydrophobic amino acids that is inserted into the ribosomal tunnel and disrupts the normal flow of translation. A commonly cited sequence responsible for stalling is RXXXXXLIVP. These tags can create a physical or chemical bond between the peptide and ribosome. This ribosome mRNA complex is used to screen for interactions with other molecules. The ribosome mRNA peptide complexes are incubated with a target protein or ligand. The complexes that didn't bind to the ligand are then washed away. The mRNA that binds with the ribosome peptide target complex is then extracted and purified. With the help of reverse transcription, mRNA is then converted to complementary DNA and amplified using PCR. And finally, the amplified sequence is then sequenced to determine the protein sequence. The applications: 1. Protein screening. 2. Design new proteins. 3. Functional studies. 4. Drug discovery. The advantages of the ribosomal display are: 1. No cell culture is needed. 2. It can handle a large library of peptides. 3. It provides a direct linkage between the protein and its encoding mRNA, which simplifies the selection process. Limitations are: 1. It requires a careful optimization of in vitro translation conditions. 2. Used for smaller proteins due to the constraints of ribosomal translation and mRNA stability. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.